So thanks for having me back. And was it really a year ago? I think that we were here. So I'll start off by acknowledging the HESI team in pulling this together, and in particular Lucilia, who's pulled these slides together. And one thing I've kind of noticed through the HESI staff that have been putting slides together, there seems to be a bit of an arms race in the amount of animations and such like that they're trying to get into their slides, which has been interesting to watch. But just to comment generally on the back of the last presentation, fantastic presentation that we had, is that what HESI does is bring people together. Convene is what it does. And bringing disciplines together is where synergies occur, and I think we saw that tremendously in the last presentation, where we had an incredible marriage of computer science and biology, and the power that was able to generate was quite phenomenal. And there's no way that computers can do that on their own, and there's no way that biology could do it on its own. So the potential within these collaborative programs is enormous. So excellent and really nice to be part of it. So this emerging idea or problem, if you like, that we put together last year centered around cell therapy. So it follows quite neatly from the last one, where the parallels and relationships were fairly obvious. Cell therapy is gaining massive momentum, or certain areas of cell therapy are, but there are still significant problems to overcome to enable it to be translated. Another part of HESI's mission, as it were, into the clinic. So we're very pleased that it's been taken on. So that obviously happened this time last year, and we had our first committee meeting by telephone and WebEx towards the end of December last year, and we've had regular monthly calls since then, gradually building up and understanding where we want to go and what we want to do. And we've got a nice timeline of what we want to achieve, or what we have achieved up to this point here, where we've now got 2 subgroups identified. So the number 3, so we've got three co-chairs in this subcommittee, and it reflects really very much the makeup of the committee, as we'll see in a couple of slides' time. But so there's myself representing industry, I guess. I work for GE Healthcare. And together with Greg Mullen last year, we put together a few slides for the proposal. And Greg's a senior lecturer in imaging biology at KCL in London. And we've also been able to recruit Mercedes Sarabian from the FDA, who provides a fantastic input and really quite essential input for considering what we're trying to achieve with this committee. So what's the membership? I told you there was animation. So 48 participants, 26 organizations, and a really nice spread across those 3 disciplines making up the committee. So we're on a good first footing. We know there's gaps that we want to fill in terms of the membership of the committee, but we've had a really good start, considering we're only 6 months into it from our first meeting. So the mission. So we want to enable the safety assessment of cell-based therapies by enhancing our ability to reliably apply analytical methods, devices, scientific knowledge, all the things that committees bring, to evaluate the distribution and fate of these cells in a patient. And what we're going to hope to do that is facilitate translation of cell-based therapies to the clinic through driving the development of tools, methods, knowledge gaps where they are, required to evaluate the in vivo safety and fate of therapeutic cells. So why do we need that? Well, for small molecules, and to a large extent large molecules, an awful lot is known about what's to happen. From the very moment a scientist in an academic laboratory or a pharmaceutical company comes up with a new class of drug, small molecule, that could hit a therapeutic target, they can enter that structure into a database freely available on the web and have a very, very good indication of what its safety profile will look like. Because we've been doing this for decades, and we've collected all that information and we know what we're dealing with. Large molecules less so, but that knowledge is building. For cells, we don't really have an awful lot. For the allogeneic, yes, we can draw on certain elements of what's been learned through large molecule development, certainly from a manufacturing perspective, because the principles are very, very similar. So at the end of the process, you want to keep the cell and not what the cell is making. But for autologous cell therapies, it's very, very different, because the starting material for each therapy is absolutely unique. So there's nothing, there cannot be anything really to go on beforehand, because it is unique to that patient. But you take it out of the patient and you do stuff to it, so therefore it's likely to change. And how is it going to change? And what do we do? How do we assess that? And that's the nut, if you like, of the problem that we're trying to address here, to provide tools and technologies to enable us to do that. So this slide is in here to kind of highlight that there are possibilities out there, there are technologies out there. And the committee does have a bit of a focus on imaging, but it's something that we need to move a little bit away from in some respects. But imaging, we believe, will have a big role to play in this. The bubbles across the top kind of indicate a generic manufacturing process for cellular therapies. And it's not hugely different from allogeneic compared to autologous. You need a starting point of cells. Now, whether that's from a healthy donor or from the patient themselves, it doesn't really, from a manufacturing perspective, starting point doesn't really matter. But all the way through that process, you have to know that that cell is maintaining the properties that you believe you harvested it for. If it's a T cell, at the end of the manufacturing process, does it still have cancer cell killing properties? If it's a mesenchymal stem cell or an iPS cell, by the time you get it to the patient, does it still have those therapeutic properties that you hope it does? And you can do that to a greater or lesser degree before you administer it into the patient. But once it's in the patient, it's really a continuum of this current process. You still need to know that once it's in the patient, it's going to do what it's supposed to do. And cell imaging technologies are available across the modalities. There are tools with which you can label cells ex vivo so you can tell where they go. And that's going to be hugely important, particularly when we're trying to direct cells to, for example, dopaminergic expressing cells for Parkinson's disease. It's very important that they locate to a very specific place in the brain to enable that function to work, to return that function. For cardiac therapy, if you inject systemically, do they ever get to the heart to actually do anything? These are questions that we don't really know the answers to yet, and there are tools around that can do that. But imaging can also bring other things too, which is on the bottom left-hand side of the slide, is there are diagnostic agents out there that can help assess efficacy. And we need to be a bit clever, I think, within the cell therapy field about how we use available technologies within clinical trials to assess therapeutic efficacy. Because the potential of cell therapy in the regenerative medicine space is enormous. But the clinical trials don't seem to be actually generating that data. And yet anecdotal evidence suggests that they should. So why not? Why can't we get those statistically significant trials? So, of the questions that we're trying to address, why is cell fate distribution important? Characterize the role, cell fate distribution, in assessing the overall safety profile of a cell therapy product. What elements matter most? Well, we need to identify which aspects of cell fate, distribution, safety assessment are the most impactful for patient safety. And this is going to vary tremendously from therapy to therapy. One thing we've very quickly learned, and even our initial discussions, is that one size is not going to fit all in this case. So we have to come up with a suite of tools. Do we have tools and technologies available already that could fit or be adapted quickly? And if not, what's missing? And we certainly want to undertake exercises to try and match what current technology and methodological approaches that are out there will fit to answer those safety questions. But not coming at this just from, I've got a really, really good tool that I think I can use in this space. It's got to match the need. And that's where I think that the multidisciplinary approach that we're taking with the regulator, therapy developers, and tool developers all sat through the same committee, we got a chance of actually making that happen. And how can we define a reliable and reproducible outcome? What is a reliable and reproducible outcome? Don't know yet. You've got to work these things through. And how much precision and accuracy is required. And this is where having the regulatory authorities on board is great, because we can put to them a series of scenarios and say, how about this? Does this demonstrate to you that this therapy is safe? Is this tool reliable and accurate enough to give you confidence to move forward with the therapy? So, the parallels with diagnostics is very simply, it's all about being confident in the test to go and make a clinical decision on it. Here, it's being confident on the test to allow that therapy through to be released onto the market. So we split kind of the work we're trying to do into three kind of columns. It's that number 3 again. Distribution, survival, and phenotype. To try and work out some workflows and some areas in which to focus for the group. And the animations has disappeared. So, subgroup 1 that we think about combined 2 areas. So that we were thinking about the point of administration as being one potential area to focus on. Then there was near-term, acute safety, and then there was the latent terms. So for a cell to be effective, its time in the body will vary tremendously dependent on the therapy. There are some regulatory T-cell, Treg type therapies where the cell may only need to be in the body for a matter of hours in order to induce its effects onto the system, or a dendritic cell therapy that will have done its job within 24 hours and then could disappear. However, there are some cells, T-cells for example, that you would expect a certain percentage of them to survive for the entire life of the patient. At what point do you stop monitoring that patient and that cell to ensure it's either A, doing its job, or B, not doing something it shouldn't be? Very, very difficult questions. That one we think is a little too tough to tackle at the moment, so we're just parking it for a little while we focus on subgroup 1, where we thought there was enough commonality to address the point of administration along with the near-term or acute safety needs. Subgroup 2 is going to focus on tumorigenicity, and this has come about through great energy and determination from our Japanese colleagues who are studying this in great detail in separate groups within the Japanese environment. And they've come to this subgroup to engage with a group that's on a more global level, to enable us to take their ideas, their framework forward, to see if we can apply it and get input from a global perspective into the tumorigenicity element. So we have 2 subgroups that are forming, or have been formed. We had our first face-to-face meeting at the start of this meeting, and plans and discussions are continuing right now with email exchanges of potential ways to move these subgroups forward, with action plans to go forward. And that's taking in terms of the point of administration, this series of questions or areas in which to focus on. And for the tumorigenicity biodistribution subgroup, a potential work plan that's aligned not only to what we're trying to achieve, but what the Japanese group has already kicked off and is working towards. So, what are we hoping to achieve in the near future? Well, we've got plans for, and we will execute a workshop towards the end of this year or maybe early into next year when we can get as many people as possible together at an appropriate venue to really take forward the discussions and put flesh on some of the action plans and to adapt where we need to. And we do indeed aim to get a publication out, a white paper, if that's appropriate, on this topic, this subject, to start really getting this out and about to understand what else we need to do and how we can approach the problem. And that, I believe, is that. Thank you very much. I'll have to take questions.
Thank you, Bill. Let me just start by saying that it's actually quite remarkable, and I would like to congratulate the group. 48 participants and 26 organizations in a matter of a year. That's really fun.
6 months.
In 6 months, rather. Yeah. Well, 1 year since we last
Since we heard from you,
right?
absolutely, yeah.
So let's open it up, to questions. Bill.
Wonderful.
Anyone?
Completely clear.
I don't have candy, so I'm sorry.
Oh,
he left the bag. I can throw this.
Just to make to use this possibility, although I'm a member of the group, just a question. For cell therapies, there's we focus on the animal data, on tumorigenicity and so on. But how has it been discussed also to give attention to potency data, the endpoints, to define a quality endpoint? What is the release test for potency? Has that given attention?
Yeah. So yes, absolutely. I think this within the field that the topic of potency, particularly at the end of the manufacturing process, is a very hot topic. Now, that's not something that we're choosing to focus on with this group because there are other elements within that, and it's a piece of work that's ongoing and sort of outside of this community. The plan was really to focus on what happens in vivo. How do you test that in vivo? How can you understand what happens in vivo? And there'll be clinical endpoints to which the potency of the therapy will be assessed. However, saying that, tumorigenicity is something that can be assessed in vitro or preclinically through a number of different methods and models. Now, how appropriate they are to cellular therapies, we need to investigate, and that's certainly something that I'm sure the second subgroup will look at. But potency at the point of administration is tough. Now, at the moment, it's done through markers of expression that would indicate that the cell therapy should be efficacious. Now, sometimes that's just the volume of cells or the number of cells that you've got. Sometimes it may be a panel of markers on the cell surface to assess whether or not they should do what they should not do. But release criteria is really focused more around acute term safety of that. So is it the cell type you think? So are there T cells in your preparation? Tick. Are they over the 60% viability? Tick. Are there no microbes in your preparation? Which in fact can only be assessed, the answer you get is several days after you've administered the product, but it's part of the release criteria. And that's it. Because at the moment, these therapies, in terms of the way, the development that they are, and the stages that they are in the developing process, the risk versus benefit ratio by that point, that suffices in terms of release criteria. Those criteria are only going to increase and become more stringent the more we learn about the therapies and the healthier the patients are as they receive the therapies.
Yeah, I think it's partially answered by is an understanding of the genome likely to form part Is an understanding of the genome likely to form a part of your safety assessment, or at least even part of the genome? So if you see certain genes being expressed or certain polymorphisms, is that going to be incorporated into the safety profile of the product, which are essentially individual products to the individual, aren't they?
Yes, exactly. And I think as a possible methodology or technology to enable a safety assessment, I think the genome, genetic expression, epigenetic expression, all these potential fields could be utilized in this space. And the expression for CAR T cells, for example, you would hope that they are expressing on their cell surface, the binding antibody that's required, the binding antibody that's required for the tumor-specific antigen, and that's definitely part of the process for sure, and that's currently done by fax. As simple as that. You just take off a sample of the cells, run it through the fax machine. Is it expressing the receptor as you would expect it to do, and that's great. But how do you know that expression continues once you put the cell inside the patient? And that's the question we need to ask in some respects. And then, potentially, genomics tools could come into it.
I wish you all the best. It strikes me you've set yourself an enormously challenging problem here.
It is a challenging problem.
Within the time frames that you look at these things, I mean, you've got to take yourselves out from the patient, you've got to engineer them, you've got to put them back in a certain period of time. Your time to do the safety assessment is going to be very small.
Is very small, which is why we believe the answer is going to be in vivo. That you're going to have very little time to assess the safety prior to administration, and that has to be enable that as the patient receives the therapy. So it's an interesting and challenging process. Ruth.
Ruth has a question here.
It's related to Tim's question, I think. I was going to ask how you go about characterising the stem cells that you're going to put in vivo, such that you can then know how generally applicable any conclusions you reach about distribution and tumor-genicity are to other stem cell type products. How broad are your conclusions?
So again, because that is such an enormous task, that's something that we're not thinking of looking at. So characterization of the cells through that manufacturing process is something that we won't be focusing on, because we cannot possibly do that alongside trying to understand what happens to the cell in vivo. So it's going to be a suite of tools and technologies. We will aim to try and identify where the gaps are that need to be developed for once these cells are returned to the patient. Characterization of IPS cells and that sort of things, that we're going to have to leave to the groups that are developing the therapy themselves, so that they understand as much as possible about their particular cell type. And then hopefully we will begin to learn what those therapy developers require from an in vivo test to tell whether or not their iPS cell is indeed doing what they expect it to do in vivo or not before it's too late for the patient.
No question, no comment. Just I want to support what you mentioned. Actually, the Japan, as you are aware, Japan has already started a clinical trial with iPS cells, so that we have already experienced several hurdles for the safety issues. So based upon such experience, we thought that it is very fruitful and meaningful to collaborate with this CT track program. That's why, Japanese governmental regulatory agencies and also the pharmaceutical companies as well as academia now very much interested in collaborating, and thereby we are very happy to provide our know-how expertise to this program, so that during this process probably we can get more integrated program, and also global. Sometimes we may get some global standard for the safety issue. That's why we are working hard, and also every month we have the telephone conference. It's a wonderful opportunity to work together. Thank you very much. I would like to thank you.
Thank you.